But guys, today we'll learn about stop and go signals. You can say stop and go signs in animal cells. Like how the cell decide when to continue the cell cycle or when to stop. Okay, it is like similar to you can say traffic lights. Okay, every animal cell has internal stop signal. Internal stop signals, which means these signal like can pause the cell cycle naturally. Can pause cell cycle naturally at some checkpoints. The cycle can only move forward like when he receive a go ahead signal. Just like the green light, we can say green light, red light. We are when we stuck at the traffic, when the red light is there, we stop, and like with the green light, we ahead. Okay, these are automatic. One key thing is there, these go ahead go signals. You can say go ahead signals carried by but signal transaction pathway. Signals transaction pathways. Repeat after me. Yeah. Why the cell stop and go? Because the cell have system. It has a monitor which checks for example like this the did the DNA uh, like for example like did DNA finish replicating or not? If not, it stop. Our chromosomes are attached property to each other like in M phase, or we can say a metaphasic plate. If the cell large enough, if everything is fine, okay, the cell cycle go ahead, give the tick, blue tick. If something wrongs, it stops in a middle way. Now, where do the checkpoint collect the information like what is wrong, what is not? Okay, they collect it from inside the cell, outside the cell. There are three main checkpoints that I talked earlier also. Three main checkpoints. Da-da-da-da-da. First one is G1, second one is G2, third one is, you guess it right, M phase. The most critical one is G1 phase. Why? Because it is called as a restriction point. Why I think I pronounced it correct or not. Restriction point. Because as this phase, okay, if the cell receives a go ahead signal, okay, the cell continue to divide like G1 to S, M to G2, S to G2, and G2 to M. Okay, division happens. However, if doesn't receive no go ahead signal, its exit cycle, cell cycle, it goes into a phase which will tell you, okay, now G naught. It is also called resting phase. We can say this is like permission granted, we can divide. If permission is denied, you should rest. Okay, like most of the human cells are in G naught phase. Why? Because like you can say like mature nerve cells, and also we can say muscle cells stay into G naught phase until they are fully developed. Until they are fully developed. Most of the cell we can say can return from G naught back into G1. For example, we can say liver cells. Like when a liver part is destroyed, okay, or like liver is damaged, these cells go back into cycle and divide and repair the tissue. Now we can say example of internal signal example we can say at M checkpoint, which means mitosis. Like the main question arises here: are the chromosomes properly attached to the spindle? Which means, at the metaphase, when the chromosome attached to the spindle, like this, okay, they form a metaphasic plate. Before before going to anaphase. Okay, this is a final check. And like after going to the anaphase, okay, cells should not separate sister chromatid until everything is properly attached. And also, if even if one kinetochore is unattached, okay, it delays anaphase. This ensures uneven chromosome distribution. I'll tell you more in detail right now. Like what really happens at the end, at ending phase of the metaphase, we can say M checkpoint. There's something called regulatory protein complex. Okay, this is not like we can say this is not a cyclin CDK complex. I told you earlier. Or CDK, yeah, complex. It is different multi-protein complex. We can say, yeah. What does this thing do? This activates an enzyme we studied. It called separase. What does separase do? It cuts the cohesion proteins which holds the sister chromatin together. Holds the sister chromatin together. And then what happens? It cuts it. Separase cuts it. Cuts cohesion protein. So what happened is sister chromatin gets separated from each other. For example, this is either wait, it's getting chromosome to half. They separated each other. Yeah, and move towards opposite poles. This led to beginning of anaphase. Now a lot of guys is asking like why this checkpoint is critical, why it is necessary. It is necessary because like each daughter cell, okay, each new generation should get correct number of chromosomes, and also errors like we can say monosomy, which means missing chromosome, or trisomy, chromosomes, trisomy, which means extra chromosomes are prevented. In simple term, we can say like M checkpoint is the final stage which ensures perfect chromosome distribution. Additional thing like we can say additional checkpoints. For example, first checkpoint we can say in S phase. What happened in this phase? Like DNA is copied, replication of DNA, copy. Okay, if DNA is damaged, it's not good. The checkpoint stops. The S phase checkpoint stop the cell cycle. Stop cell cycle, and what this leads to prevention of copying damaged DNA with on continuing with errors. And new thing also discovered like around 2014, like checkpoint between anaphase and telophase. telophase, it ensures anaphase is surely or correctly finished. It's finished correctly. Verify chromosomes are separated correctly or not. Chromosomes are separated correctly or not. Not, and also it prevents cytokinesis if if like the chromosome has some breakage or missegregation. For example, if chromosomes are not divide completely in the upper region of cell and the lower region of cell, like the cytokinesis don't happen. So prevention of, but we can what was I saying? Prevention of, yeah, chromosome breakage and missegregation. Now new thing. Other reasons why cell don't divide. First of all is if they don't divide, if essential nutrients are missing or are not found, such as sugar, amino acid, beside AA, okay, or medium components, growth components, yeah, anything. You know, they need to think, like you can say, even with the nutrients, they need to think like most mammalian cells. Mammals means animal. Mammalian cells need a thing called growth factor. It is a protein which is released by one cell, released by released by one cell to stimulate another cell to divide. Different cells need different factor. Growth factor, for example, fibroblast need different, liver cells have different, bone cells. Today we'll talk about the fibroblast also. You know, they need something called PDGF. What this stands for? It stands for platelet derived growth factor. Derived growth factor is one of the, it is made by platelets. What does platelets do? They help in clotting, help in clotting. And the PDGF role is to stimulate fibroblast to divide. What are fibroblasts? Connective tissue cells to divide. Why it is necessary? Because when you get a wound, okay, or you can or cut, what happened is platelets release PDGF growth factor. Okay, what does this do further? This divide fibroblasts rapidly and they cover the clot, which helps to repair tissue internally. That's how your body heals. Body heals then. How it is done? Fibroblast thing called PGDF receptors on the plasma membrane. I'm writing as PM. Okay, like then what PGDF bind to these receptors, okay, which activates thing called receptor tyrosine kinases. Receptor tyrosine, wait, ty I guess, yeah, tyrosine kinases, which triggers a mechanism which I'll teach you later. Signal transduction pathway. This allows the signal, we can say this allows the cell to pass the G1 checkpoint, and then what happens next? You guess it right. Division of fibroblast. Now, one more thing I have to tell you about is I've talked about the chemical signals which affect the cell division. Now, there are external physical signals which stops, or we can say affect cell division. First one is density dependent inhibition. 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 Wait. Yeah. Okay. So what happened here is, when the cells are, overcrowded, okay, they don't divide. Stop divides division. Like this was tested in the lab class. We can say, oh, for example, let's just take a container. For example, this is a container, okay. There's a cell placed here, okay. Okay, now what happened is division happens. This divides. Okay, they divide until they form one layer. Okay, this is one layer, I can say. This all is one layer. Okay, once the layer fills the bottom surface, think it as a 3D view. If it fills the bottom, like top to bottom, it is filled completely, the division stops here. Now what happened if you scoop some of the cells here? The division starts again. Okay, the cells, they are empty, like this space is empty, this space is empty, they're starting to divide again. Okay, till the gap is filled. Okay, this thing is called density dependent inhibition. Okay, like why does this happen? Okay, like when the cell touch each other, the protein called surface protein, note this, these bind together. Surface protein bind together, and this protein sends a signal to the other cell, like to stop dividing. We don't have spores, we don't have any space left, don't have any space left. And next thing is encourage dependence, which means animal cells need a thing to attach for division to happen. Like just the same container, it is container, like these animal cells attach to container and then they divide. Okay, they ensure like we are safely anchored, and it is okay to divide. If the cell is floating in the air, or no, it is unattached to like the different cells, no division will happen. Now, how density and encourage signal work together, like both, what they like, they are signal to like plasma membrane proteins, okay, cytoskeletal elements, and cell cycle control pathways. These things ensure that the cell divide only at the correct location and density at time. Okay, like why are they matter for like in the body or not? Yes, they matter. Why? They help in, or they function in embryo development, tissue growth, we can say, healing of wound that I told you earlier, and maintenance of the organs. Now, what happens like if the cell doesn't follow these rules? This leads to a thing called cancer. Cancer. How? Because they do not show in density dependent inhibition. They pile up in layers. They don't form any, like they don't form just one layer, like they pile up the layer. For example, it was a container, let's make it 3D. Okay, this container, like normal cell like pile up from the bottom. Okay, that's one layer complete, but these cells, they started to pile up and fill everything. They also don't need any support, like no anchorage needed. They just bind together, like they bind while floating. Okay, they ignore the signal. What this leads to uncontrolled growth of the cells and forms a tumor, resulting in formation of tumor. What tumor is? This is because like they have ability to transform. They transform into something which is different from the normal cell cycle. Now let's talk more into detail of that. Okay, cancer cells ignored, ignored normal growth signals. Like in healthy cells need growth factors. I like to write it GF. Now the girlfriend growth factors to divide. Okay, what are growth factors? I told you earlier. There are proteins that tell the cell to divide. Time to divide. Proteins tell the cell to divide. Okay. Now what happens in cancer cells? They divide even when growth factors are missing. So like there is a hypothesis, like they're not sure, but you know what may happen is they make their own growth factors, which gives a permanent go ahead signal. Okay, and also like they also have faulty signaling pathway, which tells them like growth factor is present even if it is not there, you know and third thing is defective cell control system, which is usually caused by something called genetic mutations, or you can say changes in the DNA. So the main thing is, cancer cell divide without permission. Cancer almost begins because like the change of genes happens. Change in genes happen. Main reason is the mutation. Okay, like these things produce the faulty proteins which don't regulate the cell cycle correctly. you know as we see in the normal cells, they stop first at G1, then G2 checkpoint, M checkpoint. Okay, in cancer cells they don't stop at all, don't stop, or sometimes they stop, random stop, not at correct checkpoints. no like the time taken for the normal cell is normal cell, I'm writing NC, they divide 20 to 50 times. Okay, then they stop, age, which mature and die. Okay, which is we can say as apoptosis, I guess. Apoptosis, which means apoptosis. Let me get the correct meaning. Apoptosis, which means self-destruction of the cells, or we can say cellular senescence. In the case of cancer cells, they divide forever. Cancer cell CC, if nutrients are provided. Nutrients are provided. The one example was performed in lab was HeLa cells. Correct spelling, HeLa cells. Okay, this was a cell of the woman named Henrietta Lacks. Henrietta, which lacks some cells in 1951. They are still dividing today also. Okay, when like the cell gain ability to divide indefinitely, no stopping, whatever means to happen or not, they've undergone thing called transformation, which behave like cancer cells. Normally, you see in the normal cells, if DNA is damaged, okay, the cell trigger apoptosis. Apoptosis, which means self-destruction of the cell, or we can say programmed cell death. Cancer cells ignore this process. Damaged DNA, I'm like ding D, continues to replicate or divide. The cancer become more aggressive, abnormalities accumulate together and finally form a tumor. Cancer form from one single abnormal cell. This is resistant to immune, which means this is not destroyed by immune system. It divides uncontrollably and forms a mass. This is called mass of cells called tumor. There are two types of tumors. Now study tumors. There are two types: benign, and second one is malignant. This is type of tumor like which has start at location, original location, we can say original location it does not spread, and it is not that dangerous and can be removed. The second thing, malignant tumor. This is a thing that people scared from, called cancer. Why? This can spread and like can invade, attack on nearby tissues, can spread all over the body, all over body parts, and can cause organs, harming the organs of the body, which are life threatening, and these are true cancer cells. Now, new thing is like how malignant cells behave abnormally. First reason is, they have abnormal chromosome number. Second thing is altered metabolism, which means they do not function as normal tissue cells, do not function as normal tissue cells. Second, loss of adhesion, which means sticking. They do not stick together like anchoring to divide. Okay, do not. Okay, which this thing allows to invade nearby tissues. And fourth thing is the next thing called, this helps in inducing angiogenesis, which means making new blood vessels. The cancer cell CC send signals to make new blood vessels grow toward the tumor, which supplies the nutrients. This supplies nutrients, and which resulting in fast tumor growth, faster tumor growth. The next thing is even more scary. I'm talking with the process called metastasis, which means spread of cancer. What happens in like some tumor cells, okay, like they detach, they get first they are connected, like they're disconnected from the original tumor and enter into blood vessels or lymph vessels, travel new part of the body, travel new part, and form a new form new tumor. This is metastasis, and it makes cancer more than we can say deadly or dangerous. For example, in the mammary gland in females, this is a mammary gland. Okay, yeah, this thing is called a glandular tissue. Okay, and this thing is red. Okay, forming in the tissue is called as tumor. Okay, now what happens is this try to multiply here and there. They invade the neighboring tissue. Okay, and then what happens is they spread to the like blood vessels and the lymph vessels. If, for example, this red one is this one, yeah. This red one is the blood vessel, writing as BV, and green one is the lymph vessel. Okay. Now small amount of cancer cells, or we can say tumor cells, pass through them and metastasize the another part of the body from these blood vessels and the lymph vessels. This results in metastasis completely. Okay. Now let's talk about some treatments. First thing is called as radiation therapy, which is used for localized tumor, which are placed at a specific point in the body that we know, okay, which damage the DNA. Okay, cancer cell cannot repair the damage, can't repair the damage, and unfortunately, they die. In normal cells, however, what happens in normal cells? Normal cells can do DNA repair, so they survive. The next thing is chemotherapy. What happened is you know this thing attacks, when the cancer is metastasized, which means like these things mainly attacked on active dividing cells, which are present all over the body. What happened is, for example, a drug called Taxol. Okay, what does this do? It freezes the mitotic spindle, you know prevent microtubules from depolymerization, prevention of prevents microtubules from depolymerizing depolymerization, and then what happens is they'll get stuck in metaphase, which leads to death, of course. But this process is, it is very harmful. The chemotherapy is harmful and expensive. Why? It has side effects also, as you know, like the chemotherapy attacks all the fast dividing cells. Okay, but this medicine doesn't know like which one of the healthy and which one are the bad cells, so they also attack healthy cells also, healthy fast dividing cells, which leads to thing called nausea. Okay, hair loss, and weakens immunity power get weakens. As you know, like immune cells also divide very frequently. Hair follicles also divide very frequently and very fast. And nausea because intestine cells also divide very quickly. So like there is not like correct medicine like that completely remove the cancer, but yeah, it can get less, which because of the chemotherapy. Yeah.